Hi, my name is Ellis, and my research was based on investigating the protective effects of green tea extract on melanoma cells. Now, melanoma is one of the deadliest skin cancers known to man. It causes about 60% of all malignant skin cancers, and UV radiation is one of the major melanoma causing agents. Sunscreens that are supposed to protect us from harmful UV radiations do not have any therapeutic effects on already damaged cells. So, in search for a promising therapeutic agent, green tea was discovered. Green tea has one extract called epigallocatechin 3 gallate, which has shown to have promising therapeutic effects in reducing UV-induced skin cancer. Thus, we hypothesized that green tea extract EGCG will attenuate UVB-induced cell damage and downregulate some of the tumor-promoting genes. To test this theory, we first had to establish a sublethal UV dose. We used A375 cells, human melanoma cells, and we also used a UV radiation gel box to irradiate our cells. We had a control group of untreated cells, and we irradiated our cells at 5, 15, and 30 minutes respectively. To measure the cell death, we used a cytotoxicity assay MTT using MTT tetrazolium salt. Then, after establishing the sublethal dose, we irradiated our cells, and immediately after, we dosed them with some green tea extract at 25 and 250 micromolar with a vehicle treatment of UV only. We then had two outcome measures. One was to measure cell state right here, and the other one was to measure gene expression. To measure cell states, we used the acridine orange ethidium bromide staining, where green cells were live, yellow cells were apoptotic, and red cells were necrotic. Then to measure our gene expression, we measured two genes, H2AX and COX2. H2AX is a gene responsible for DNA damage, and COX2 is a gene responsible for inflammation, which is a form of tumor promotion. We predicted that UV radiation will cause DNA damage and cell death. In line with this prediction, we found that 5, 15, and 30 minutes compared to the control had significant reduction in the cytotoxicity. Now, we also wanted to study the synergistic effects of UV and DMSO. In our DMSO only positive control compared to our UV time points at 30 minutes, there was significant cell death. Further investigation used a 5-minute time point as our sublethal UV dose to study the cell states and to study the gene expression. Surprisingly, we hypothesized that apoptosis will be reduced as in a dose-dependent manner with green tea. However, we found that there was a reduction in necrosis at the 250 micromolar treatment, but this was just a trend. There was no significance here. Coherently, we also found that there was an increase in the number of live cells, the percentage of live cells, when we increased green tea to the 250 micromolar. There was a trend. Surprisingly too, we found that EGC treatment had no significant change in gene expression. That was especially surprising for COX2, since we expected that UV radiation induces COX2 expression. Perhaps there was no inflammatory stimuli present, which is also surprising and unexpected. H2AX is a DNA damaged gene, and we expected it to be highly expressed. But since there was no significant change in the expression, it's suggestive of the fact that H2AX is not a reliable indicator for DNA damage. We were successfully able to discover a sublethal dose of 5 minutes, which resulted in about 70% cell viability. We found that there was no significant change in apoptosis, but there was a trend of decreased necrosis, which correlated to a trend of increased live cells. There was also no significant change in COX2 or H2AX expression compared to the untreated and UV vehicle controls. It is recommended that future studies with larger sample sizes and maybe ELISA or immunofluorescence with the H2AX protein would lead to better understanding of the mechanisms underlying green tea's effects on cell states, inflammation, and tumor promotion. I would like to take this opportunity to thank my supervisor, Jessica Livingston, and my other lab technicians and friends.